Maybe I get started. Good morning, everyone. Welcome to another Terasaki seminar series. Today we have Professor Chiara Gabbi. She is a medical doctor specialized in internal medicine and PhD in medical science. She works as a physician at Humanitas Medical Care in Milan, Italy, and she is also affiliate scientist at Karolinska Institute in Stockholm, Sweden. Originally from Italy, she spent a large part of her career abroad. First at Karolinska Institute, where she obtained a licentiate degree and a PhD in medical science. Then in USA, where she was a research assistant professor at the University of Houston and scholar at Harvard Medical School. She has authored many scientific papers for innovative activity. She received many international awards, including the Standout Woman Award in 2018. Besides her work as a physician and scientist, Dr. Gabbi has been very active in training and supporting junior scientists. In particular, she has designed and personally lectured in more than 30 research institutions to about 1,000 junior scientists. The floor is yours.
Thank you, Professor Dokmechu, for the introduction, and thanks for having me with you today at the Terasaki Institute. I'm really honored and grateful for the invitation and for the opportunity of showing you my research data. So the title of my webinar today is Hormo-metabol-genomic, a novel paradigm for personalized prevention and therapeutics. So during our webinar today, I will show you how hormonal, metabolic, and genomic factors could interplay together in determining disease susceptibility and disease progression. We will start from the hormo-metabolic part, talking about nuclear receptors, in particular a receptor called the LXRB receptor beta, and I will show you how hormonal and metabolic factors during particular genetic condition could orchestrate in the pathogenesis of gallbladder cancer. From cancer, we will go into genomic, and I will show you that there is a particular group of individuals that have a high risk of developing COVID-19, and also they have higher risk of early death. At the end of the webinar, I will reveal you what is the molecular link connecting hormonal, metabolic, and genomic factors, and I will tell you how in the future we could target this molecular link in order to design personalized approaches, both for prevention and therapeutics. So let's start from the hormometabolic part, and let's jump together into a cell where we find the nuclei of cells and where we find nuclear receptors. What are nuclear receptors? Nuclear receptors are intracellular receptors. They bind to small biologically active molecules, for instance, cholesterol-derived oxysterols, fatty acids, vitamins, steroid hormones. There are 48 members of the nuclear receptor superfamily, and each member has its own specific group of ligands. So they work both as intracellular receptor and as transcription factors. So when nuclear receptors are activated by specific ligands, they bind to a coactivator complex. They work usually in heterodimerization with other nuclear receptors and quite often with a receptor called the retinoic receptor. Together, these two nuclear receptors, they bind to the promoter of specific target genes, and in turn, they can regulate gene expression of particular genes, of particular pathways of genes. So, if you think about the ligands of nuclear receptor, you can immediately understand how, for instance, nutrients from the diet, what we eat, the content of fatty acid, the content of cholesterol that is in our diet, can have a direct effect on gene transcription. So, regulating what we eat, we could have a direct effect on gene transcription, and in turn, we could regulate multiple biological processes like metabolic homeostasis, reproduction, development, and differentiation. So One of the members of the nuclear receptor superfamily is called the liver X receptor beta. And actually my whole scientific career has been focusing on liver X receptor beta. LXR beta is strongly expressed in epithelial cells, in particular in cholangiocytes, in the ductal cells of the pancreas, in the lung, in bronchial cells. And it is also expressed in neurons and glial cells. Activators and ligands for liver X receptor beta are cholesterol-derived oxysterols. In particular, these 4 oxysterols, they are ligand activators of LXR beta. There are commercially available also 2 synthetic ligands for research purpose. However, these ligands have low affinity for LXR beta. They can also activate the twin brother of LXR beta, LXR alpha. And LXR alpha, when active, could have some side effects like fatty liver. So one of the challenges I'm working on now is to identify specific LXR beta ligands. LXR beta works in heterodimerization with the retinoid X receptor, and when it's active, there are 4 main biological pathways that are regulated by LXR. The first one is cholesterol export. LXR beta works as a sort of sensor in the cell. So when the cholesterol concentration in the cell gets high, cholesterol is transformed into oxysterols. Oxysterols bind to LXR beta and induce, and LXR beta in turn induces the transcription of cholesterol exporters in order to prevent cholesterol accumulation inside the cell. Together with cholesterol export, LXR beta controls water transport, and also it has an anti-proliferative action and anti-inflammatory action. So when LXR beta is active, we have cholesterol export, water transport induced, reduced cell proliferation, and very importantly, we can reduce inflammation. I just told you LXR beta is strongly expressed in epithelial cells, and here is an example from the gallbladder. The gallbladder is a small organ under the liver storing bile that is a key really for digestion. So here we are in a gallbladder, and you can see in brown there is the expression of LXR beta in the nuclei of cells, both in animal models and in humans. So all the brown nuclei are nuclei that are immunoreactive for LXR beta. In order to understand the role of LXR beta in the gallbladder, we used animal models knockout for LXR beta. And here is what we found. So first of all, I show you a normal female gallbladder. You can see here that the normal epithelia is a nice, elegant monolayer of cells, one beside the other, well organized, the normal gallbladder. However, when we saw the gallbladder of our LXR beta knockout animals, you can see the picture is completely different. We found that at 11 months of age, those mice had dysplasia, metaplasia, and adenoma. So these are preneoplastic lesions. When mice got old, 19 months old, actually, we found carcinoma in situ and locally advanced carcinoma. What was really surprising to us was not only this phenotype, but also the fact that LXR beta knockout male gallbladder were perfectly normal. You can see here, we follow the mice in all the stages, 11 months, 19 months, and we couldn't find any neoplastic lesion. Actually, they have something else that I will tell you at the end of the webinar. So this specific female phenotype is something seen also in humans. Actually, gallbladder cancer has an incidence that is 2 to to 6 times higher in women compared to men. And importantly, hormonal replacement therapy in postmenopausal women can increase the risk of gallbladder cancer. And this is a paper we just published. It's a collaboration I have with Dr. Koshal and Dr. Jackson at the NIH. And you can find more details in the paper I'm citing. So gallbladder cancer in human associates with the female sex, with hormonal replacement therapy, and also with cholesterol gallstone disease, metabolic disorders like obesity, or a dietary intake rich in carbohydrate. This interesting fact that there is an association both in animal models and in human, an association female sex hormones and gallbladder cancer, led us to a key research question. So what about taking out, shutting down the hormonal signaling that comes from the ovaries? Are we going still to see neoplastic lesions in our mice? So we performed ovariectomy when the mice were 3 months old. We followed the mice till the age of 11, 12 months. And what we saw is this. Well, the sham animals, sham animals, they still have the ovaries. They had this metaplasia in the gallbladder, so preneoplastic lesion. We found that there was an upregulation of the TGF-beta signaling, leading to a loss of E-cadherin, that is this cell surface molecule that keeps cells adherent one beside the other. You can find more information in our paper published in PNS. So this is the picture of the sham animals, and to our surprise, our hypothesis was valid. To our surprise, actually, LXRB ovariectomized animal did not show any preneoplastic lesion, and E-cadherin was expressed back on the cell surface of the cholangiocytes. So we could prevent gallbladder cancer development by shutting down the hormonal signaling coming from the ovaries. And then we ask ourselves, okay, what about LXR-beta in human gallbladder cancer? After our paper was published, a study from India based on our preclinical evidence was published, and here is the study in which 2 genetic polymorphism of LXR-beta were identified associated with female, specifically female, gallbladder cancer. So there is a particular genetic profile related to LXR-beta that is associated with gallbladder cancer in humans. We went then to see samples of patients affected by gallbladder cancer and tried to see where LXR-beta was. So in normal epithelia, you can see that LXR-beta is expressed in the nuclei of cells, while in adenocarcinoma, we see LXR-beta in brown, also expressed in the cytosol of cells. So there is an immunoreactivity that is, in some samples, more prevalent in the cytosol, while in the normal epithelia, where we have LXR-beta prevalent in the nuclei. So here is a key point. I told you LXR-beta is a nuclear receptor active in the nuclei of cells because it works also as a transcription factor. So if LXR-beta is in the cytosol, it means that probably it's not doing his job of transcription factor. So here is another paper showing that actually in the top 10 canonical pathways that differ between gallbladder cancer and normal gallbladder, the pathway regulated by liver-X receptor beta is the one that is mostly changed, and actually is the one that is downregulated in gallbladder cancer, meaning LXR-beta is not transcriptionally active. And you can find more details in the paper I'm citing. And now another key question came up. What is the link then between cholesterol, since LXR beta is activated by cholesterol-derived sex hormones, and the pathogenesis of gallbladder cancer? So here is a cartoon for you. We are in a cholangiocytes. You see there is LXR beta in the nuclei. LXR beta is activated by cholesterol derived and induces the transcription of cholesterol transporters. These transporters, in particular ABCG5 and ABCG8, they promote the elimination of cholesterol from the cell. So if we do not have LXR beta in the nuclei, what happens? Well, we don't have LXR beta and we have then a lack of transcription of genes encoded for cholesterol transport. And if we don't have enough expression of these transporters, cholesterol may accumulate in the cells. And here really it is exactly what happens in our animal model. Here is a PCR in which you can see that there is a downregulation of the cholesterol transporters, ABCG8 and G5. There is an upregulation of the LDL receptor that picks up cholesterol, and also there was an upregulation of the enzyme synthesizing cholesterol inside the cholangiocytes. So downregulation of the cholesterol transport, what happens is an accumulation of cholesterol. And here again, we are in our animal model. You can see in the upper panel the LXR beta female gallbladder, in which in red, this is an oil red staining, you can see all these lipid droplets red, and on the right you see a bigger magnification of the square in the left panels. When we check the normal gallbladder, there are some small red dots. This is the physiological level of cholesterol. And you can see the difference. There is cholesterol accumulation in the perineoplastic lesions. So cholesterol and cancer, this is now a key topic in oncology. Cholesterol accumulation is not something peculiar of gallbladder cancer. Numerous papers are coming out showing that actually in the neoplastic microenvironment of breast cancer, of GI gastrointestinal cancers, renal, thyroid cancer, there are high level of cholesterol accumulating in the microenvironment of the tumor. And also it has been shown that there is a deregulation of all the enzymes and transporters involved in cholesterol homeostasis. Importantly, there is an important evidence showing that dietary factors, for instance, a food rich in saturated fat, and I would say rich in oxysterols, are associated with higher incidence of numerous cancer, and conversely, treatment that reduce cholesterol level could be effective in hindering cancer progression in some cohort of patients. Then the key question is, how could we put together metabolism, cholesterol, hormones, and carcinogenesis? And here is another cartoon, a model for you. So in normal condition, we have again LXR beta that is perfectly working, activated by oxysterols and inducing the export of cholesterol. There is another nuclear receptor called estrogen receptor alpha that is activated by the sex hormone 17 beta estradiol, the female sex hormone. ER alpha has a pro-proliferative action. So when it's active, induces the transcriptions of gene mainly involved in proliferation. You may have heard for sure about ER alpha in breast cancer. So what happens when we have an impaired LXR beta signal? Well, we have, you know now very well, that we have an accumulation of cholesterol derived. For instance, 27-hydroxycholesterol. And 27-hydroxycholesterol that accumulates can bind to ER alpha. It is an activator of ER alpha, can bind to ER alpha, and then promote the pro-proliferative action of estrogen receptor alpha. So patients that have probably a genetic polymorphism that reduces the activity of LXR beta, and that at the same time eat a high-fat diet, they have high risk of developing cancer, given that we have these three factors together, a diet rich in, for instance, cholesterol, a reduced LXR beta signaling, and then a possible activation of ER alpha. So this is the working hypothesis connecting really metabolism, hormones, and a particular condition in which we have LXR beta that is not fully functional. So thinking about a vision for personalized medicine, we can really use gallbladder cancer as a model of hormo and metabolic cancers. In designing therapeutic approaches, but also preventive personalized approaches. So for instance, we could use LXR beta genetic polymorphism in screening individuals that have high risk of gallbladder cancer. Who are these individuals? Well, females. You have seen in women, we have a higher incidence of gallbladder cancer, but also obese patients or people that are used to have a high diet rich in saturated fat. So we can screen them in order to determine the risk for gallbladder cancer, and then design effective preventive approaches. We can use LXR beta nuclear localization both in the diagnosis and in the follow-up or in determining the prognosis, because if LXR beta is in the nuclei, it means it's working. If LXR beta is mainly in the cytosol, well, it means it's not working properly. So we may have cholesterol accumulation, and we may have an activation of the GF-beta pathway and all the pathways that I just showed you. LXR beta selective agonists, when they will be available, could be tested as adjuvant together with the standard of care in the treatment of patients with gallbladder cancer. And also how much ligand we have available could be used in trials to determine the prognosis, follow-up, or recurrence risk of patients that are at high risk. So this is a new view specific for hormo-metabolic cancer and specific for patients that have a reduced activity of LXR beta. So from cancer, we go into genomic, to the second part of our webinar, and we talk about a disease that unfortunately we all know very well, COVID-19. So as of yesterday, we had more than 6 million deaths in the world. And since we really got used to numbers, and unfortunately sometimes it's difficult now to understand the magnitude of what is happening, well, 6 million people is about the population of Missouri or Colorado or Finland or Denmark in Europe. So a huge crisis, but I'm convinced that out of crisis there are opportunities. So what is the opportunity that COVID-19 is giving us? Well, I'm convinced it's personalized medicine, and I will show you now why. Here is the outcome scale of the World Health Organization released at the beginning of the pandemic. Really, since the early stages of the pandemic, we saw that patients had different clinical pictures. So there were patients that were perfectly fine at home, asymptomatic, and other patients that underwent death, and sometimes even early death. So I'm going to show you now data from the GeneCOVID consortium. It's a consortium of more than 30 Italian hospitals. I'm a part of the GeneCOVID consortium. This GeneCOVID consortium is led by Professor Ranieri at the University of Siena in Italy. So we have 874 patients that were enrolled in the GeneCOVID study from March to June 2020. What was evident was, beside the high clinical variability of the COVID-19, it was really evident that the clinical severity was not related so much to the age of patients. Of course, patients that underwent death, they were older, and patients at home were younger. But then in the hospitalized categories, we saw, for instance, that patients in the intensive care unit undergoing invasive mechanical ventilation had a mean age of 60 years. And they were younger compared to patients still in the hospital, but with low flow oxygen. They had a mean age of 65. So age was not so much a determinant of severity of COVID-19. And the key question that we had really since the beginning is are there genetic factors that could determine COVID-19 progression? The answer is yes, and one of these genetic factors was identified by the Gene COVID Consortium. We identified 40 patients in our cohort that were more prevalent in the category of patients receiving invasive mechanical ventilation, and they were also more prevalent among patients younger than 50 years that were in the intensive care. These high-risk patients are patients that are carriers of a single pathogenic variant of the gene called CFTR. What is a CFTR? CFTR is a gene that encodes for a channel, cystic fibrosis transmembrane conductance regulator. It's a channel expressed on the apical membrane of epithelial cells that transports chloride and bicarbonate outside the cell. So when there are more than 2,000 mutations, pathogenic mutation described in the CFTR gene. So when the CFTR gene has a pathogenic variant, the channel sometimes cannot migrate depending on the type of mutation, sometimes cannot migrate to the cell surface, other times migrates, like in this example, but is unable to work as a transporter. So what happens is that the secretions, they become very sticky, causing plugs and obstruction. And here is an example of the very sticky mucus that we have when CFTR is not working properly. Besides working as a chloride and bicarbonate channel, CFTR has other biological function. It is involved in regeneration, it is involved in differentiation and polarization, it is involved in development, in epithelial to mesenchymal transition, and also in cancer. Dysfunctional CFTR could promote cancer progression. So it's a channel with many other jobs. So thinking about what happens when the channel is not working properly. So when we have both copies of the gene encoding for CFTR that have loss of function variants, so we have both copies carrying loss of function variants, there is a disease that develops called the cystic fibrosis. Probably you have heard about this disease. Is a severe disease, autosomal recessive, affecting children and young adults. It's a multi-organ disease because CFTR is expressed in many organs, lung, pancreas, liver, intestine. And this disease, cystic fibrosis, characterized by pulmonary insufficiency, pancreatic involvement, leading to respiratory failure and quite often premature death. So how is the inheritance of cystic fibrosis? Here you can see in this cartoon, in yellow, there is the pathogenic variant, and in gray is the normal variant. So in order to have a kid with two pathogenic copies of the gene, variants in both copies of the gene, we need to have two parents that are carriers. So there is a severe impairment of CFTR in cystic fibrosis, and there is a mild impairment of CFTR in the carriers. So those carriers are people that have a good general health, but they have higher risk, as I just showed you, of developing severe COVID. And I want to show you more data on these carriers and COVID. Actually, when the carriers undergo invasive mechanical ventilation in the intensive care, they have a mean age of 51 years. So they are much younger compared to the non-carriers who undergo ventilation at the age of 61. So these are young people ending up in the intensive care because of COVID. Also, they have a higher relative risk of reduced pulmonary function, respiratory function, and they have a higher relative risk of having a high inflammatory response. So the inflammatory markers in the blood, like the C-reactive protein, are much higher compared to the non-carriers. And what happens? Well, they have a severe COVID, but also in our court, they had a higher risk of early death. And for early death, I mean a 14 days death. The hazard ratio for 14-day death was 3.10 after adjusting for age, sex, and comorbidities. And it was significantly higher compared to non-carriers. You can find more details on our study in the paper that I'm citing. So just before the pandemic came out another paper that I'm citing here, showing that actually these carriers not only have higher risk, of course, of COVID, as we know now, of severe COVID, but also they have higher risk of many other diseases affecting organs in which CFTR is expressed. They have high risk of respiratory infection, of respiratory disease, of pancreatic disease, of pancreatic cancer, of gastrointestinal disorders. And thinking about the magnitude of all these diseases, here I'm showing you the daily score, the disability adjusted life year. This score represents the loss of the equivalent of 1 year of full health. So you can see how many years of full health could be associated with the status of a CF carrier. And in other words, what I'm showing is that if we see the burden of disease in the world, well, actually 6 groups of diseases could be related to the CF carrier status. What about then the impact of the CF carrier status in the population? What is interesting is that the prevalence of cystic fibrosis varies among ethnicity. For instance, it's higher in Caucasian compared to African American or Asian. When the CF carriers were screened in the European Union, it is estimated, it is known that 1 out of 25-30 inhabitants are CF carriers, resulting in a projected population of about 15 million European CF carriers. So an important or relevant number in terms of, let's say, global health. So thinking about then the impact of CF carriers in the different ethnicity, and thinking about that since we have a different incidence of cystic fibrosis among ethnicity and therefore countries, we thought, okay, let's see if the CF carrier prevalence that we obtain by measuring the number of CF patients in each country relates somehow to the COVID prevalence or the COVID case fatality rate. So we did a study, ecological study in 37 countries. And here I really want to acknowledge the work of the foundations in Europe, Canada, and US that registered the data on CF patients. So we used the prevalence of CF patients as an indicator of the prevalence of CF carriers, because in order to have one patient with cystic fibrosis, we need to have 2 parents that are carriers. So here is what we saw. There was a direct correlation between the prevalence of CF carriers or of the prevalence of CF and the prevalence of COVID-19 in the 37 countries studied. And the association, the direct correlation, was maintained also when we weighted the results for the population. The size of the marker relates to the size of the population in each country. There was the same correlation also when we waited for the number of tests performed. And these are data only of the first wave of the pandemic. When we study the correlation CF prevalence and COVID-19 case fatality rate, we saw again a direct correlation. And the correlation was maintained also when we weighted the results for the population. So the CF prevalence, the carrier prevalence in these 37 countries relates really to the prevalence of COVID and also to the case fatality rate. So what happens then to this carrier, going back from population to individuals? What is the model of disease development? Well, there is this is of course a model, but I think it's two models of disease development in which we have two or more than two hits that reduce CFTR function. The first hit is genetic and is the presence of a single CF-causing pathogenic variant. And then during the life, these individuals undergo other hits, SARS-CoV-2 infection, estrogen exposure, arsenic exposure, smoking, they all reduce CFTR function. And then they end up having higher risk of, for instance, severe COVID-19, pancreatic cancer, and all the CF-related diseases that I just showed you in one of my previous slides. So everything is a balance in terms of CFTR function between the factors that induce a gain of function of CFTR. There are also gene modifiers of CFTR that can increase the function of CFTR. There are CF drugs, but there are also loss of function factors, genetic, but also environmental factor like smoking or arsenic exposure. So here is a key point, and I'm guiding you towards the end and the revelation of what is the molecular link that I told you at the beginning. So we thought, okay, if we can increase, since we have one normal copy of the gene and the other is pathogenic, could we increase the expression of the normal one? And the key question is then, what is regulating CFTR? Because if we know what is regulating CFTR, then we can induce the expression of CFTR. And here is the first step. Guess what is the key transcription factor regulating CFTR? Well, it's our friend liver X receptor beta. This is a paper we published last year in which we identified LXRB beta as a key regulator of CFTR in the gallbladder, a key inducer of the transcription of CFTR in the gallbladder. And actually the answer came from the male gallbladders. I showed you at the beginning that the male gallbladders did not undergo carcinogenesis, but actually the male gallbladders were much bigger compared to the wild-type gallbladder. They were much bigger, and when we check the gene expression, actually there was a reduction in the water channels, because as I told you, LXR beta also controls water channel. You can see the PCR here and the immunohistochemistry on the right. In normal condition, aquaporins are expressed on the apical membrane of cells. LXR beta male animals did not express aquaporin. And very interesting, also CFTR was downregulating both at the mRNA level and at the protein level. So LXR beta could be really a key factor regulating CFTR. And then we are back to our visionary slide. So what is the vision for CF-related diseases? The list of the diseases that I showed you, but also severe COVID-19. So while we can use absolutely CFTR pathogenic variants, so we could sequence people in order to see if they have higher risk. But also there is a sweat test that is done in cystic fibrosis patients. Is not not the standard in carriers. We need to measure chloride and bicarbonate in the sweat. I know at the Terasaki you have sensors, so this is something interesting that could be done. If then we have a patient that is carrier, how could we prevent the development of diseases? Well, a trial with drugs increasing CFTR function could be done. If we have already a patient, let's say with pancreatic cancer, we could sequence CFTR and use CFTR drugs, CF drugs as adjuvant therapy in trials testing the efficacy of these drugs. And also the CFTR functionality could be a prognostic marker or something used in the follow-up and recurrence of patients affected by CF-related disease. So a visionary slide that shows you really how we could design specific individualized prognostic, therapeutic, diagnostic approaches based on a particular profile, of LXRB and CFTR. So this is the conclusion of my webinar. I said I will tell you what is the molecular link. Well, I think there is an axis, and this is what I'm working on, the axis LXRB CFTR, that could be really the link between hormonal, metabolic, and genetic factors in disease susceptibility and progression. And this could be the target for personalized approaches, both in prevention and in therapeutics. And finally, I want to thank all my previous, current collaborator, friends, and co-workers in Sweden, in Italy, at the University of Houston, where I previously worked, and a really special thanks to all the founders that supported the studies that you have seen. And with this, I really thank you for the attention, and I'm open to take any questions you may have. Thank you.
Thank you for this great presentation, Dr. Gabbi. The question came in, what percentage of gallbladder cancer is caused by LXR beta mutations?
Well, still, this is something that should be done. The paper from India was from a small cohort, and actually we don't know still how much of this gallbladder cancer is really caused by variants of LXR beta. It's something that requires big data, and is something that we are working on. So at this time, I cannot answer the question that is a key question, because then it would lead to the magnitude of the LXR beta related cancer. So it's something in progress. But you can have an idea on the prevalence in India reading the paper that I cited.
Thank you. Thank you. Let me see. One other question is, you mentioned about hormones. Our sweat sensors can also measure cortisol levels, other stress factors from the sweat. Do you think that there could be a correlation between cancer progression, drug therapy, and these cortisol levels?
Well, absolutely could be. Absolutely could be. And actually, there is another nuclear receptor, the corticosteroid receptor. I haven't mentioned this nuclear receptor in my presentation, that is activated, regulates also the stress response, regulates inflammation. Yeah, there could be absolutely a role of glucocorticoids in the pathogenesis of cancer, absolutely, through the specific nuclear receptor.
It's a very sophisticated presentation you had, and I see multiple axes, the hormonal level, surface receptor, genomics. Do you use machine learning type of things to analyze your data?
Oh yeah, this is something that I really would like to do as a next step, because I'm really convinced that these are huge challenges. I show you the burden of disease. We are talking about cancer, that is the biggest challenge ever, COVID-19. So we need big data. Absolutely. We need big data, population data, and we absolutely need artificial intelligence, machine learning in order to really understand what patients are at risk and in order to understand what is the best treatment. Regarding the COVID-19, with the Gene COVID Consortium, we used artificial intelligence to identify variants that have high risk, that are associated with a high risk of COVID-19. And you can find the paper in PubMed, Gene COVID Consortium. We identify common and low frequency variants of many other genes associated with severe COVID. So if you put in PubMed, Gene COVID Consortium, you can find the paper in which we used artificial intelligence. But just to conclude my answer, yes, we need absolutely to get together to physician, engineers, expert in artificial intelligence in order to really understand personalized approaches for this type of diseases.
Thank you. Another question is, you picked cystic fibrosis, even though your approach, the hormono-metabolic-genomic approach, can be utilized in attacking other diseases. Is there a particular reason you picked cystic fibrosis?
Oh, wow. Oh, I love this question. A really good point. Well, I haven't told you, and I was hoping for this question. Actually, there are a lot of analogies between LXRB being not functional and cystic fibrosis. Cystic fibrosis patients, there are reports that both patients and carrier can accumulate cholesterol. So they accumulate cholesterol in the cells, a phenotype similar to a low activity of LXRB, as I show you. And also cystic fibrosis has a different clinical presentation, let's say, between male and females. Females patients, actually, they perform worse than male patient. In one of my slides, I show you actually the survival for male is about 40 years, for females is a little bit lower. So there is also a role of hormone that needs to be understood in cystic fibrosis. There is a role of metabolism in cystic fibrosis also, because these patients, they accumulate cholesterol. So we picked the cystic fibrosis because of the analogies, and also really to us was a key, the paper that I show you, when we saw this huge gallbladder in male animals. And we knew that a huge gallbladder was characteristic of cystic fibrosis animals. And then we measured the transporter, and then based on the analogy on the phenotype and based on the molecular finding, then we reached the association. So cystic fibrosis is could be also considered a disease that has hormonal and metabolic, specific hormonal and metabolic profile.
Excellent. Well, that's all the questions I have. Thank you for your time and giving
thank
a
you
great
so
seminar.
much.
Appreciate your time. Thank you.
Thank you so much again for having me with you, and I hope next time to be in person with you at the Terasaki. So thanks really for the invitation.
Thank you.
Thank you. Have a good day.
You too.